Calvin cycle. Okay, itu je. Okay, what that is the basic idea or basic mechanism in the plants, in the light reaction. Okay, so whatever notes that you read, okay, there are many facts, there are many details about the light reactions. Okay, in the end of the day, it will go back, it comes back to the principle that I've just explained you today, right? All right. Okay, see, products from the light reaction are free energy in the form of ATP and NADPH. Okay, to produce ATP and NADPH. So remember. Okay, so in the case of electron that are in the case of ATP that are being produced, it's not enough, right? Because ATP is needed for the Kelvin cycle, right? If the production of ATP is not enough, right? So there is a situation or a cycle where the system will recycle the electron back to the first electron acceptor here. Okay, so see these are the destiny or the journey of the electron. Okay, right, all right. At the F and R, all right. Instead of the transferring electron to the NADP plus to NADPH, it's the FNR will go back and transfer the electron here, right? So remember last time, every times plastoquinone, okay, these proteins, okay, accepting the electron, it will bring together the protons from the stroma here, right, to the lumens. Okay, so we know that after they produce the ATP using the ATP synthase, right, the protons will be more concentrated in the stroma section. Okay, so they pump in into stroma, right? So every time they pump the hydrogen, the protons to the stroma sites, it will produce ATP. Okay, but in the case of the ATP is not enough, right? So the electron will be transferred back to the plastoquinone here, right? So plastoquinones will transfer ataupun will reduce the cytochrome B F6, so the journey of the electron continue, and then at the same times the hydrogen will be pumped in, from the lumen to the stroma, pump out, from the stroma to lumen, pump in. Okay, so the protons will be pumped in back here, so the concentration here will increase, and then the cycle continue, right? They akan pump out the protons and produce the ATP, right? So the principle, the result is the same, producing the ATP. All right. NADPH is a molecules that cannot be generated simply like this. Okay, the NADPH need to be produced. If the NADPH is not enough, so the system will have to start gene expression. That is different story. It's not a molecules that can be generated from the physical production like ATP. All right, that needs ATP synthase, right? The ATP, if plants want to increase the numbers of ATP, this is the different story, tapi related. All right, kalau the system want to increase the ATP, right, it can also increase the number of protein ATP synthesis, but it will also involve the gene expression, right? If the ATP synthesis is enough, the machine is enough to produce ATP, will not involve any gene expression, right? You know that gene expression is the production of ATP transcription producing the mRNA, and mRNA will be translated into proteins, and then proteins depending on the gene that has been expressed. Okay, I'm not sure kalau saya dah try explain a bit on the molecular punya base. I'm not sure. I think that was the most simple punya explanation. I think you have learned it in the foundation in biology punya class, right? Right. So stop there. Okay, stop here. Penghabisan cerita for the light dependent. Okay, simple je. So now we go to the dark reaction. A bit more complicated, but not easy to be. I know it's not hard to. It's more complicated, but not difficult to understand. Right. So okay, we got equation here: 6 carbon dioxide, 12 water, light energy, carbohydrate, which is glucose, and 6 oxygen, and 6 molecule of water. I repeat, I don't remember actually the equation, right? But I can tell you why is it 9 carbon dioxide, why is it 12 molecule of water, why carbohydrate, and 6 oxygen. To produce 1 molecules of glucose, it needs 6 carbon dioxide, right? Because the structure of the glucose 6 carbon. That's why it needs 6 carbon dioxide to form a molecule of glucose, right? So so when it's and then the rest of the equation is just to balance lah, right? So ada 6 oxygen, right? Become 12, right? Balance with the 12 here, right? So the first calculation to balance adalah based on the fact that to produce a molecules of glucose needs 6 carbons. Okay. Okay. Calvin cycle is a series of complex reaction that takes part in the stroma, right? The stroma section contains more enzymes, right, than in the lumens. Okay, lumens are basically the place where the protons are accumulated, right? The lumens are a place where the water are being split off, right? Being split off, produce the protons and produce the oxygen. The oxygen will go freely, all right, until something happens. But in the normal condition, the oxygen will go freely, right? The oxygen will be used back in the respiration, or mostly they akan release as a free oxygen to the atmosphere. Okay, and then the protons will stay in the lumens, right? When the concentration is high enough, so they will be used to produce ATP. Remember this diagram. Okay, so the stroma side is where the Calvin cycle happens. Calvin cycles involve a series of enzymes. All right, the carbon dioxide, when they are being fixed, all right, by an enzymes, it's not literally transform into a glucose. No, it's not simple as that. It needs a series of conversion, all right? It needs series of formation that involve different enzymes, okay? For example, just after the carbon dioxide it fades, right? And then with, there will be, for example, nanti kita tengok, go for the next reaction. Let's say got C 5 carbons. This is the original molecules, all right, original proteins, plus one molecule of carbon dioxide. This carbon will be used, all right, to finally form acid glucose, right? So ada, so means that ada 6 kali lah, okay, memerlukan tiga, this what we call RUBP, ribulose 5 biphosphate. Ada 5 carbon, so dia akan fix 1 carbons, alright, menjadi 6, alright, jadi 6 carbons, right? So dia akan split off, and then after 6 carbon dia akan split off, become 2, right? So every times ada changes, right, dia akan involve enzyme, right? So these are enzymes that are important in the Calvin cycle, right? So these enzymes dan cycles or metabolic activities, right, happen in the stroma, because the stroma punya area place is very suitable. It's like our cytoplasm. It's also similar to cytoplasm part in the plants, where it is an aqueous environment, right, holding 100 and probably 1,000 enzyme in the cytoplasm itself, right? So the stroma is like that, right? The environment is suitable for the metabolic activities to happen, okay? Any metabolism lah, right? So that is stroma, okay? Calvin cycle happen in stroma because of the environment. The lumens a difference, okay? Carbon fixation from atmosphere converted into compounds, okay, occurs during these cycles. The cycle use ATP and NADPH from light reaction and carbon dioxide from atmosphere, right? So again, the cycle use the ATP and NADPH from the light reaction, and the carbon dioxide they catch during the light reaction. During the light reaction pun boleh, during the dark, the fixation of carbon dioxide can happen at any times, right? During the day, okay, ataupun during the night, because the fixation of carbon dioxide doesn't need light. That's why the dark reaction or Calvin cycle, it is called light independent. Tak ada light pun tak apa, right? Okay, there are 3 processes in the Calvin cycle that are important. I don't expect you to remember all the steps in the Calvin cycle, because for me, as long as you understand what is the photosynthesis, that should be okay. Like I don't expect you to remember 3 phosphoglycerates, because I don't remember the first product is 3 phosphoglycerate. That's why I tell you, that's why I wrote here number 2, because I don't remember. Okay. But the process you have to know, there are 3 that are important. What that is: carboxylation, reduction process, regeneration. Right? Carboxylation is the carbon fixation. Right? Carbon from atmosphere. All right. Go into the stomata. All right. And then the carbon dioxide will go into the mesophyll part, and then will be captured by ribulose 1,5 biphosphate. Okay. Okay, first dia di captured by an enzyme, the important enzymes. This enzyme is the most abundant proteins in the world, that is rubisco. All right. Carboxylase oxygenase means that it can fix, okay, carbon dioxide. It also can fix oxygen, right? And then rubisco helps in the carbon dioxide fixation, and then dia akan transfer lah kepada ribulose 1,5 biphosphate menjadi 6 carbon molecules, alright. And then this molecules this molecule is not stable, right? 6 carbon molecules not stable, and then dia akan berpecah menjadi dua, or break down into 2 molecules of 3-phosphoglycerate. Okay, settle. Carboxylation. Alright. Okay, those 3 carbon ni, dia akan go for the next reaction. We call it reduction. The process of accepting electron for 3 carbons molecules. Okay, why I said 3 3 carbons or 3 PGA, 3 phosphoglycerate is the same. Okay, sometimes I use interchangeably the names of the molecules, because the 3 carbon can also phosphoglycerate, can also be pyruvate, can also be some other types of molecules, right? But in general, we can use 3 carbons or 6 carbon molecules or 5 carbon molecules. Like ribulose 1,5 biphosphate is a 5 carbon molecules. So I can just say 5 carbon molecules, right? So for this stage is 3 phosphoglycerate is 3 carbon molecules, right? And then 3 carbon molecules ni akan go for the reduction process. We go for the reduction process, the process of accepting electron, and also with the help of ATP. This ATP and NADPH are coming from the light reaction. So that's why they need, Tuhan dah jadikan cantik, right? The process needs ATP and ADPH, so that Allah SWT menjadikan another set of process, what we call light reaction, to produce ATP and ADPH to feed up the process of reduction, right? So after NADPH transfer the electron, reduce the reduced 3-phosphoglycerate here, all right? So dia akan losing the electron, so NADP plus will go back to the light reaction to accept a new electron from the light reaction, and then the process continues, right? Dia sangat cantik lah dia. To produce NADPH, to produce NADPH, right? It needs electron, right? At certain points in the electron transport change in the light reaction, right? Dia akan pump in protons, right? So bila dah proton dah got too concentrated, right? More concentrated than the other sides, right? So dia akan produce ATP. So both elements here will be used back in the carbon cycle, right? So ATP akan jadi ADP plus PI. So this also will be likely go back to the light reaction area, right, where ADP and PI ini inorganic, phosphate yang tidak bercampur, any elements yang tidak bound to any molecules, kita pakai inorganic, like as a phosphate and elements, alright. That's why you learn one of the importance of having phosphorus, right, in a mineral nutrition. One of it adalah ni lah, ATP and ADP, right? So ADP akan during the reduction process, ADP will be broken down, becomes ADP and phosphate inorganic, and energy release. So energy release ni will be used to reduce or will be energized the reduction process. Okay, so ADP and phosphate in organics will go back to the will be substrate again to produce another set of ATP. Okay, so bila ATP synthesis here, right? ATP synthesis here, right? See, the substrate is here, right? So protons will go up and then turn the turbine, spin the turbine to produce energy, and this energy will be used to be stored and to be used and then stored in the ADP in the ATP punya compounds. So bila ATP dipecahkan, energy release. So where does the energy coming from? We know that energy cannot be created but uh, can be transformed, something like that. Good. They spin, they transfer the energy by the ATP. Right? ATP will be used uh, to fuel up the process of reduction, right? And then the energy tadi akan hilang as a heat, right? So these are the reduction. The process that involve ATP and ADPH is a reduction process, right? And then the three carbon tadi akan go another transformation and finally produce triose phosphate. The apa ni? The paling basic molecules to form sugar, right? Triose phosphate, right? So triose phosphate akan produce sucrose. Sorry, triose phosphate akan produce glucose, and then glucose can form sucrose and then more complex carbohydrate, which is starch. All right. So after dia tukar, after sugar has been produced, all right, the 5 carbon, okay. So macam ni, Kelvin cycle to form an one molecule of glucose, it needs 2 cycles, right? Means that every cycle to it needs 3 molecules of this one, right? To fix 3 carbons to produce triose phosphate, which is also 3 carbon, right? And then so bila carbon tu dia dah combine, okay, carbon dioxide dia akan ribulose 5 carbons and then plus 1 carbon 6, right? So blah 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 go for the akan go through a few modification and conversion and formation, right? So dia akan attach, right? The 5 original carbons of ribulose 5 biphosphate ni dia akan go back and fix a new carbon dioxide. Okay. The production and also rearrangements of the ribulose, because first dia okay because dia 5 carbons, there's a 6, right? And then dia akan pecah dua, dia akan pergi their own way, this one also go for their own way, and then dua-dua dia akan release 1 carbon, alright, dia akan jadi apa, jadi to produce glucose lah, right? And then bila dah berselegak macam tu, dia akan go back to process we call regeneration. So carbon tadi akan berlaku je formation balik untuk membentuk the original molecules ataupun proteins we call ribulose 1,5-phosphates to accept a new carbon dioxide. So ni ada akan apa? The process continuous. We call regeneration. And the regeneration process also it needs ATP. Okay. Tapi even though it involve ATP, dia tidak melibatkan apa-apa. It doesn't involve any electron transfer. That's why kita tak panggil process reduction ataupun oxidation. Right? Here involve electron transfer from electron carrier, right? That's why it is called reduction process. All right, ATP ni just to fill out the formation. All right. Okay, so sorry, saya kena buat restatement balik. Okay, in normal condition, when ATP broken up, okay, dia akan membentuk ADP plus ATP. Alright, so this will be used back in the process of the production of ATP. Okay, a certain but for this process, sorry to make you confused. Okay, for this process, ATP, all right, is to be used to fuel up the reduction process, and at the same time to attach one phosphate into the carbon molecules to make glyceraldehyde triphosphate. Okay, so in many cases they akan broken down ADP plus phosphate inorganic, and phosphate inorganic ni dia akan randomly moving around until dia di combine balik until it is formed back to form ATP, right? At some is many cases, okay, many cases in the metabolic activities in our cells, right? Some products, all right, some molecules need to be activated by adding phosphate, all right, by adding phosphate. So addition of phosphate here mediated by ATP. Okay, so ATP akan supply phosphate, right? Dan mana tahu nanti you go and you tengok study balik Kevin cycle. Okay, if you want to do the revision of Kevin cycle, you suddenly ini datang mana phosphate ni, alright? You remember yang saya cakap phosphate ATP after broken down dia akan ADP and menjadi phosphate inorganic, and then we go moving around, right? So nanti bila tengok structure dia, mana phosphate ni datang, itu yang saya clarify. Ada there are processes of the breaking down the ATP and produce ATP plus phosphate inorganic, and also got a reaction that ATP dia akan the P tadi dia akan attach to the molecules, so yang tinggal ni hanya P saja. Okay, ATP to ADP plus PI is just equation in chemistry. Okay, P ni boleh attach kepada molecules and also can moving around. Okay, it just ATP becomes ADP and PI to just for the sake of the chemistry punya equation. But in the real life, in the real reaction, ATP can go and attach to the molecules and also will be ataupun digunakan balik, recycle back to produce ATP. So settle carbon cycle, tak ada banyak masalah. So these are the simple things, and the rest of the notes is just explanation kepada the figure that I've just explained tadi lah. Right? This is the RUBP, ribulose 1,5 biphosphate. Right? Why 1,5? Because of because biphosphate, ada dua phosphate here attached to carbon number 1 and carbon number 5. Right? So 5 carbon backbone. Alright? And then attach, and then they accept. Okay, ni sebagai general lah. Okay, 6 carbon dioxide plus 6 carbon of ribulose 5 phosphate. Okay, dia akan jadi 6 molecules of carbons and then broken down, right? Okay, this process is yang dipanggil, it is called as a carbon fixation, right? And then go for the reduction. This is the more details of reaction, right? So for example, PGA to BPGA, alright, mula-mula ada PGA 1-phosphate, alright, 1-phosphate ni, this phosphate originally coming from the ribulose 1,5-biphosphate, alright, attach 1 ATP, jadilah biphosphate, biphosphoglycerate and bi ada 2. Right? What I explain here is just for your understanding, alright. I don't remember all this reaction, like I said every times. I don't remember. I don't remember what PGA is, right? Tapi I can explain. This is something that I want you to adapt lah, ataupun you can get. You don't have to remember, tapi you tahu mana every reaction that happens. You can explain. You don't have to remember, but you can explain. Alright. So don't worry too much. The question will not be so much detail as you can see, as you are seeing in the slide, right? Kalau saya nak tanya pun, saya akan bagi the fact. For example, saya tak akan bagi you tinggal, I will not give you to be thinking like, okay, BPGA ni apa? Saya akan give the fact, okay, the terms here. For example, if the molecule here is biphosphoglycerate, I will put biphosphoglycerate. Okay, the rest is just whether you can explain what's process, okay, how the process of making the PGA or BPGA, something like that, right? But every molecules that happens involved in the cycle, you don't have to remember, because saya pun tak ingat, right? So PGA ni nama dia phosphate, right? Macam mana datang kata phosphate? Because of ada phosphate di sini, the first 5 backbone, alright? Because of ada 2 phosphate here, got 2 phosphates here, right? So that's why it is enough to produce 3 molecules with 1 phosphate each attached to the molecules, alright? So 1 get each. Each get 1 lah from the ribulose biphosphate, alright? So bila ada ATP, the addition of another set of phosphate becomes biphosphoglycerate. And then the byproduct is ADP. ADP will go and find another phosphate in organic to form ATP, right? Alright. And then akan ada NADPH, and then NADPH join in, alright, akan berlaku rearrangements of the biphosphoglycerate, right? Dia akan jadi glyceraldehyde 3-phosphate, okay? So dia akan jadi 3 carbons molecules attached to 1 phosphate only and remove 1 phosphate. Phosphate, this one phosphate is phosphate in organic, alright? Phosphate in organic, dia tak ada tempat lain dah phosphate in organic ni, right? The most and very likely future reaction for the phosphate inorganic is to form ATP with ADP, alright? Settle. Alright, and then dari 2 molekul ni jadi 6 carbon or glucose. Ini yang datangnya the first reaction, right? Kalau you faham the cycle, if you faham the mechanisms, right? You tak ingat pun, you tak hafal pun, tapi you know how the system works. And the end of the day, you tahu, alright, kenapa, then you will understand kenapa 6 carbon ni kat depan. Why 6 carbon? Is that the random number the scientist just put there, right, just to balance the equation? No, the answer is no. The answer is because to produce 1 molecule of glucose needs 6 carbon dioxide, right? Katakanlah, right, from sejak azali, from the beginning of times, alright, photosynthesis process will produce 5 carbon molecules, right? So this number would change to 5. I do not know, probably 5, right? It needs 5 carbons to produce one, alright, 1 molecules of carbohydrate designed to the photosynthesis process or carbohydrate production. Simple. Settle. Settle. The same things. These are more details. You don't have to remember. Tapi it's good for your knowledge, right? Why I say this good for your knowledge? Because I think I've told you last week that photosynthesis process is a very important process, right? Regardless any subject or any research or any experiments the plant scientists in the world are doing, in the end of the day, indirectly or directly related to photosynthesis process. Okay. Sebab why do we need to perform experiments? Why do we need to study plant science? Of course, we want to increase the yield. All right, increase the yield, increase the plants and growth. All right, because of what? Because of to feed up the whole population in the world lah. Right? So bila kita cakap to increase, when we say about increasing the yield, about increasing the plant and growth, the main process that define that growth, define that production of yield is photosynthesis. Okay? That's why I said photosynthesis is very important. Okay. Setakat ni okay tak?
Okay.
Okay. Saya dapat tahu yang apa daripada Dr. Razik, plant physio is salah satu subjek yang diperlukan dalam mentomenti lah, sebab dia sangat susah. Betul ke? Korang memilih fisiologi sebagai subjek yang susah kan? Okay. Sebab dia kena banyak menghafal or because of the way I teach? Right. So my part, don't worry, you don't have to remember many things. As like, okay, I'm going to do the final exam. Right. Dia macam saya cakap tadi, ada few general terms that you have to understand lah. You have to remember, right? Ada three stages. Okay, carbon fixation. Okay, benda lah ni logic. This is very logic, right? Because of number one, for example, to make a glucose, you need carbon dioxide. But how do you want to produce? How the plant is going to produce carbohydrate? Because carbohydrate in the molecule form. Tapi normal carbon dioxide is moving around the atmosphere. Okay. Of course, akan kena ada proses. Of course, there is a process to capture the carbon dioxide from the atmosphere and then bind them into the molecule punya forms. Okay. Number one, of course lah, dia kan ada proses what we call fixation. Alright? Ataupun carboxylase, sama je. There is an enzyme that fix the carbon dioxide, we call ribulose, nama enzyme tu saya tak ingat lah. Ribulose 5-phosphate carboxylase oxidase. Rubisco. Oh, sekejap, saya kena google. Saya nak ingat. This is very important enzyme, tapi saya tak ingat. Tapi I know the apa? Okay. Okay. So nama dia ribulose 1,5-phosphate carboxylase oxygenase. You can call this carboxylation. Carboxylation is a carbon fixation. Sama je. Fixation, carboxylation. The terms derive from the name of the enzyme, carboxylase, alright, and oxygenase. Carboxylase pengikatan enzim carboxylase adalah the fixation of carbon dioxide. Is at the back refer to the molecules and enzymes, alright. Of course, that so bila dia capture, it's not simply as the carbon will go in and then fix into glucose. Of course, there is a process yang saya just mentioned, carbon fixation ataupun carboxylation. Alright. Bila ada carboxylation, alright, dia tak ada energy. So the energy coming from the ATP, right? We know that glucose kan ada is a molecules of carbohydrate. Carbohydrate got energy in the molecules. So we have two things again. Where does the energy coming from? All right, where does the energy coming from? Carbon dioxide tak ada carbon dioxide has no significant energy in the carbon dioxide itself. All right. The proteins of the ribulose 1,5-biphosphate is a molecule that has no significant energy in that molecules. And then suddenly, how does the glucose contain energy? Tu yang kena fikir, right? So the energy coming from the electron carrier. Electron carrier got energy from where? From the electron. The electron got energy from where? From the light dependence. You know, that's why energy is important, right? To convert, all right, go back to the definition, the conversion of light energy to the chemical energy, right? Chemical energy store in the form of glucose, right? Itu namanya process of reduction, right? The transfer, the ATP and also the electron carrier ni high energy electron carrier, right? They transfer the energy to form glucose. So the energy is conserved in the glucose, right? After the production of glucose, okay, the proteins that involved in the carbon fixation need to be arranged back so that they can accept a new set of carbon dioxide. So we call regeneration of ribulose 1,5-biphosphate sector 3. Saya tak cerita pun pasal 3-PGA bagi PGA, apa benda pun saya pun tak ingat. Any enzyme pun saya tak involve langsung. But at least that is the way that you can understand. Sebenarnya proses carbon dioxide ni tak adalah the process of Calvin cycle ni tak ada dah difficult mana pun. It's not so much difficult as we thought. I learned Calvin cycle during my foundation in matriculation and very stressed. I do not know. I think the teacher dia tak tahu the principle of apa yang dia ajar. So bila dia ajar, dia just go for the notes lah. Alright. So for this, you don't have to remember the enzyme ke apa ke, because I remember when I give it was a question about Calvin cycles, and the student very generous enough. My previous student is very generous enough for every step in the Calvin cycle to produce the names, right? Which is good. Tapi you takkan gunakan pun after you graduate, right? Apa yang you gunakan adalah the general terms tadi. Ada fixation, ada reduction, ada regeneration, right? And then during the stress, okay, during the unfavorable condition, right, keadaan yang tak okay, the condition is not okay, environment is not okay, because plants cannot run, they just stay there for the whole life. So whatever happens in the environments, they need to adapt, they need to switch, okay, they need to acclimatize, to the environment so that they can survive. All right. All right. In the in favorable environments during, for example, the temperature is very high. All right. We know that rubisco here, you see the rubisco is an enzyme, right? Ribulose biphosphate carboxylase oxygenase. It can bind both carbon dioxide, it can bind the oxygen also, it can bind. If it binds to the carbon dioxide, it will produce the glucose, all right? If it binds to oxygen, all right, it will not produce the glucose, but it spend it will spend the ATP, tapi dia tak produce any glucose, right? It doesn't produce any carbohydrate. We call photorespiration, a respiration during photosynthesis ataupun light uh, photorespiration during photosynthesis. Ni photosynthesis and respiration lah. During carbon fixation, they also bind the oxygen, right? So instead of having carbon dioxide to produce 6 carbon molecules were eventually broken down into 2 molecules of 3 carbons, if they bind to the oxygens, it will produce 3 carbons. It still stay 5 carbons, tapi still unstable and be broken down into 2 molecules of 2 set molecule of carbons and the arrangement. Okay, satu 3-phosphoglycerate, satu lagi dia akan produce phosphoglycerate. Dia akan produce the carbon dioxide. Okay, so dia akan of course carbon dioxide yang dihasilkan here, produce here, akan digunakan balik untuk produce carbohydrate in the Calvin cycle. Tapi it's like macam it's like you spend and then you get it back, the same things. Okay, you lose 1 carbon and the same carbon you make the carbohydrate again, right? The equation is still zero lah, right? Equation will be good if the rubisco bind the carbon dioxide from atmosphere, from external carbon dioxide. All right, so 2-phosphoglycerate finally will go back to 3 carbon, right, molecules for the regeneration process. Tapi dia akan consume ATP, so maknanya membazir tenaga. You waste the energy tapi you tak produce carbohydrate. In the normal condition, if the enzyme fix the carbon dioxide, right, you consume the energy and you sebagai result, as a result you produce the glucose. So you gain something lah when you spend your money and then you get something, right? For the photorespiration, you spend the money tapi you tak dapat apa-apa. Okay, because of the process what we call photorespiration, because of the limitation of the enzyme. Because enzyme ni dia tak mengenal sesiapa, dia just bind oxygen and just bind carbon dioxide. Kalau you increase the concentration of the carbon dioxide, means that you increase the chance of having the glucose being produced lah. Right? Kalau the environment that times oxygen is more concentrated, then you will get photorespiration and you will get nothing. Okay, now ini last quote. Now we go for the type of the photosynthesis process in plants. We got C4, we got C3, and we got acid carboxylation, something like that. Citric acid cycle. Alright, ada 3. C3, C4, acid granulation. Alright, ataupun citric acid punya, I think citric acid kot. Right. Punya cycle. Alright, we go for the C3 first. We go to C4. Boleh kat sini. C3. Yeah. Okay, tak boleh. Okay, kita I tell you on the C4 first. Right. Okay, C4, C3 is a method of how plants have its own system to reduce the photorespiration process. Right? How they can exclude the photorespiration process. Right? For the C4 plants, it has two types of mesophyll cell. Two types of cell we call mesophyll cells and bundle shaped cell. The one for C4 and C4 plants. Okay, we know that the Rubisco enzyme can fix both carbon dioxide and can fix oxygen. For the C4 plants, okay, there is another cycle before the carbon dioxide reach to the Kevin cycle, right? Carbon dioxide. All right. Alright, remember kita for the C4 plants ada dua section, mesophyll cells and bundle cell. Alright, so for the ni C4 C3 okay, for the C4 plants, okay, on the mesophyll cells, in the mesophyll cells ada 1,3 carbons we call phosphoenol pyruvate. Not sure you got here or not. Pyruvate ni because of 3 carbon. Pyruvate 3 carbon. Pyruvate is 3 carbon. Phosphoenol ni the arrangement of the, ada kumpulan phenol. Alright, ada group of phenol dalam 3 carbon tu lah. Right? Alright. So, phosphoenol pyruvate. Right? Phosphoenol pyruvate is a molecule, and all phosphoenol pyruvate also have enzyme. Alright? Phosphoenol pyruvate carboxylase is an enzyme like Rubisco. Alright? Cuma phosphoenol pyruvate ni, saya short form jadi PEP lah. Alright? PEP ni hanya they only can bind on the carbon dioxide, not oxygen. That's why PEP enzyme is called PEP carboxylase. Alright? Kalau dia boleh, if it can bind to oxygen, the name will be different lah, would be phosphoenol carboxylase and oxygenase. But because of the enzyme can only fix on carbon dioxide, So nama dia PEP. So the name is PEP carboxylase. Right. So the PEP only can the PEP can only bind the carbon dioxide to form oxaloacetate. Four carbons. Okay. See, if there is a chance of having, of course, other chance because it's mesophyll cells, right, directly after the stomata, directly under the stomata, all right, the oxygen or carbon dioxide will go into the mesophyll cells, all right. For the C4 pathway, in the mesophyll cells, all right, ada PEP carboxylase. It's not Rubisco, tapi ada PEP, but PEP carboxylase. It can bind only carbon dioxide. So, there is no chance of oxygen being fixed. All right. So PEP will change to oxaloacetate, and then oxaloacetate will be converted to malate. All right. Malate is still 4 carbons, and then malate ada channels. All right. Got channel here. See the arrow, right? The malate can not transfer, can go through a channel and then masuk dalam bundle and go into the bundle shaft. Alright. So malate will go into bundle shaft and then it will release ada enzyme. There is an enzyme lah that make the the release of that contribute to the release of the carbon dioxide from malate. Right. Malate will release one carbon dioxide and then automatically after losing one carbon dioxide become 3 carbon, the nama dia pyruvate. Right. Pyruvate will go up and enter the mesophyll cell with the half of ATP. Alright, jadi phosphoenol pyruvate again. Alright, so here Rubisco will start functioning here. Alright, so in this in the bundle shell, oxygen tak dapat masuk. There is no oxygen can go penetrate into the bundle shell cell. So the only substrate that Rubisco can fix is carbon dioxide. Because of what? Because of the precursor of the reaction in the mesophyll cells. So the reaction in the mesophyll cells helps the Rubisco to accept only, to fix only the carbon dioxide, right? The oxygen can only play around here, cannot be transported here, right? So that the PEP in the mesophyll cell can only in the mesophyll cell, the PEP can only fix the carbon dioxide, change to oxaloacetate, and then change to malate. Malate can go through the channel, enter the bundle shell, and release one carbon dioxide and become 3 carbons molecules, and then it will be changed back to PEP and then start new fixation of carbon dioxide. All right. So after the release of the carbon dioxide can be fixed to Rubisco, no competition with the oxygen. And then the normal reaction start here. Okay, that is C4. For the C3, there is no bundle shaft in the section. All right. So in the C3, right, in the mesophyll cell, all right, the chloroplast in the mesophyll cells, right, the oxygen will go in as well, right? The Rubisco, there will be competition between C, carbon dioxide, and oxygen for the active sites in the Rubisco. So photorespiration only happens in the C3 pathway. There is no photorespiration happens in the C4 because of the mechanism here. They are filtered out, right? They filter out already the substrate that can be bind with the Rubisco, right? Tapi ada setback of the C4 plants, right? C4 pathway, right? The PEP carboxylase, it needs a threshold temperature to be activated. I think you probably have learned in the biochemistry in the class, enzyme is temperature dependent. All right, either minimum temperature, optimum temperature, or maximum temperature for enzyme to be working properly, ataupun start working, right? In this case, PEP carboxylase, it needs like more than I think 30 degrees something, right, to activate it optimally, right? Optimally. If no, carboxylase ni will be less efficient, right? Data efficient, or it cannot, even the carbon dioxide coming in because of the temperature is not very very kind to the enzyme, so it cannot be working efficiently to fix the carbon dioxide. All right. So in the low temperature area, right? C4 plants is less efficient than C3, even though C3, right? C3 plants, right? The Rubisco, the carbon dioxide will compete with oxygen on the active side of the Rubisco, tapi but the chances of carbon dioxide to be fixed in the Rubisco and finally enter the Kevin cycle is still there. Still got chance to make carbohydrate, right? Even though there is a competition between C2 carbon dioxide and oxygen, right? But in the C4 plants, because of the low temperature, the enzyme is not activated properly because of the temperature. Even though the numbers of carbon dioxide, even the efficient of the system, right? Carbon dioxide cannot be fixed because of the handicaps ataupun the weakness of the carboxylase enzyme of the PEP carboxylase, right? But for the tropical climates where we got high temperature, daily high temperature, C4 is better than C3. It's efficient than C3. Okay, so like just imagine kalau pokok padi is C3 plants, all palm is C3 plants. Just imagine if Tuhan jadikan all palm and padi are C4 plants. Okay, we will theoretically, right, more yield will be harvested. Okay, contoh of C4 plants is corn. Alright, sebab tu that's why corn is not very favorable for growing a big plantation in the temperate country, negara sejuk. No, right? That's why if you go and see country that grow corn is Mexico, US, the southern part of US, not the northern part of US, southern part of US, for the Australia got also got area, the temperate area and also high temperature area. So they grow on the high temperature area, right? Because the corn can survive with higher temperature and more efficient photosynthesis, right? Without photorespiration, right? So for the low temperature, C3 is better. For the higher temperature, C4 is better. So, and the final is CAM, crassulaceous acid metabolism. Okay, CAM pathway is like C4 plants. Okay, it's like C4 plants. It just C4 plants as C4 plants do the photosynthesis during the day, and also light reaction happens during the day. Okay, for CAM, right, it also has two anatomical structure. Sorry. Okay, sorry. For this CAM plants, they only have one anatomical structure, which is mesophyll. They don't have bundle shell, right? Tapi in the mesophyll, there are the two separate section, alright? Two separate section. Let me check whether it's wrong. Let me double check. Oh yeah. Right. So in CM, there is only one anatomical structure which is mesophysis cells. All right. Tapi dia ada dua separate section, which is the same reaction happens in the C4 plants, which is they have got PEP carboxylase, okay, change to oxaloacetate and malates and other organic acid, and then with and then dia akan apa? Dia akan release the carbon dioxide. Organic acid ni organic acid is a organic compound lah, yang ada carbon dioxide. Sorry, yang ada carbon. Organic compound is organic where the carbon is the backbone, right? So dia boleh release the carbon dioxide or it can be attached ataupun it can form with a new carbon dioxide, alright, ataupun new carbon, right? So in this CAM pathway, dia akan release the carbon dioxide, so carbon dioxide will be concentrated and have higher chance to sit on the active site of rubisco. It just in the CM pathway, mostly pokok cactus plants in the desert, right? They cannot open stomata during the day, means that they will lose more water, right? What they do adalah during the night, alright, during the night, uh, during the night, normally photosynthesis stops, okay, because there is no energy. Alright. So what the plants do adalah during the night, they fill up, they capture as many as they can to capture the carbon dioxide. Alright. And they store the carbon dioxide in the form of malate or other organic compounds. Alright. They store sets up in the molecules. Alright. So they akan just harvest the carbon dioxide, right, from the atmosphere, but there is no photosynthesis process or the process of making carbohydrate because of there is during the night lacking of the energy. So when during the day the stomata close, but they have got reserve of the carbon fixation during the night. So during the day, malate or other organic acid will release the carbon. There is a reaction, of course. There will be another story how the carbon dioxide will be released, involve enzyme as well. But the idea here is the malate or other organic acids. Okay, malate is also one of the form of organic acid. You can call malate or malic acid. So malate or other organic acid will release one carbon dioxide. So this carbon dioxide will enter the rubisco, and then during the reduction process, the ATP will come in, the NADPH will come in, all right, to mediate the reaction in the Calvin cycle, right? During the day, the stomata closed, tapi got reserve of organic acid that contain carbon dioxide, okay, as a result of the carbon fixation during the night. So during the day ada light reaction, okay, during the day in the CAM pathway, the light reaction still occurs, okay. The light reaction doesn't need to have carbon dioxide or no. The light reaction is the light reaction is independent to what happens in the Kelvin cycle in general speaking. Okay, kalau more details dia akan, of course, dia ada other relationship. Okay, so for example Kelvin cycle, okay, Kelvin cycle said, okay, we need more ATP, it will tell to the light reaction to produce more ATP, right? That is some sort of communication. But in general speaking, all right, the light reaction happens or not is independent to what happens in the Kelvin cycle. What happens in Kelvin cycle, I don't care, right? During the day, got light, so light reaction will happen. Produce ATP, produce NADPH, all right? So, Kelvin cycle ni pula dependent to ada substrate or not, all right? The Kelvin cycle is dependent to got substrate or not, got carbon dioxide or not, got ATP or not, got NADPH or not. If there is no, there are no sign of having carbon dioxide, Rubisco, ATP and NADPH, the Calvin cycle will stop. All right. So during the day, all right, light reaction still happens even though stomata close, right? Right? Stomata close or open has got nothing to do with light reaction, right? Stomata close or stomata open has a very big effects on the Kelvin cycle because the Kelvin cycle needs carbon dioxide from the atmosphere, right? If the stomata close, so dia tak ada substrate of carbon dioxide. That's why the CAM plants during the day they close the stomata, during the night they harvest the carbon dioxide, and then bila they harvest the carbon dioxide, they fix the carbon dioxide, tapi photosynthesis process, which is carbon cycle and light reaction, are not happening. So they have to wait until the next morning so that the light reaction will start, a Calvin cycle will start as well. So then the other organic acids or molecules will release the carbon dioxide, and carbon dioxide will fix into Rubisco, and then the cycle continues. That is the CM pathway. C for plants only happens during the day. Okay, you can see from here. All right, look at the number one, productivity. Even though CAM have got very efficient, a bit more efficient than C3 pathway, but you know, the CM plants normally grow in the desert. You know, desert soil structure is not very good, soil chemistry is not very good, the mineral contents are also not very good, right? Of course, the production very low, all right? So C3 is moderate, C4 is high because of the system that I've just tell you, right? The optimum temperature here, the optimum temperature 30 to 40 cm35 ni normally desert memang, of course lah, high temperature, right? See here, 15 degrees we consider low temperature, all right? That's why in the low temperature, as I told you, that in the C3 plants is more efficient than C4 plants in the low temperature because at the 15 degrees, the photosynthesis still happens. Even though there is a competition between carbon dioxide and oxygen, but the process is still happening. There is still chance of carbon dioxide fixed into the Rubisco. In this case, if the temperature doesn't reach this one optimum temperature, the optimum temperature is the best temperature. The minimum temperature probably 25. The minimum temperature is the minimums, the lowest temperature that can not induce the activation the activation of the enzyme. In this C4 plants is PEP, carboxylase. So it needs higher temperature to activate the carbon dioxide, the enzyme to fix the carbon dioxide. So that's why in the lower temperature, even though they have a very significance of significant amounts of carbon dioxide, they could possibly fix, but because of the inability to be activated by the lower temperature, so the plants cannot be functioning very well. Okay, sorry. I made a mistake here. Bundle cell like chloroplast. Dia ada bundle shell, alright. Ada two section anatomical structure. alright? Tapi have got low chloroplast. So photosynthesis does not happen here. Okay, very little, right? But scientists, for the diagrams to facilitate and make it easier to explain, okay, sebab tu dia letak dalam ni. Satu ni je. Sebab Kelvin cycle happen here. Right? Kelvin cycle happen here. Right? Because the chloroplast very low in the Van der Shelf. Tapi in the C4 pathway and the mesophyll cells adalah site di mana hanya berlaku the carbon fixation by the PEP carboxylase and oxaloacetate and change to malate only. Alright. Okay, ada kekeliruan antara tiga pathway? Got any confusion? Because we are ending our lecture. So any confusion or any question on the blood reaction ke, carbon fixation ke, or three types of these three pathway? Semua dah faham ke? Oh, ataupun tak tahu apa nak tanya?
Tak tahu apa nak tanya.
Okay, tak tahu apa tak tanya tak apa. Tapi yang sebelum, yang lain faham?
Faham.
Okay. Kalau tak ada soalan, okay, any question on the lab section tak? Okay, tak ada juga. Mana-mana lab lah. Sebab kan saya just bagi arahan from the WhatsApp kan. So if there is any confusion ke, there's something that you don't understand ke. Okay, kalau okay, tak apa. Okay, saya nak buat random check. Hafizan ada kat sini? Hafizan, are you there?
Yes, doktor.
Syairin ada kat sini?
Ada doktor.
Right. Farisha ada kat sini?
Ada doktor.
Right, good. So kalau tak faham, kalau semua dah okey, this is benda yang paling simple, not fundamental tapi paling simple lah saya boleh explain for the for the topics of photosynthesis. And next kita akan masuk for the respiration. Respiration tu pun kalau sekali pandang tu pun ingat macam payah lah sebab ada cycle right? Tapi tak sepayah tu pun, right? Macam Kelvin cycles, tak perlu pun hafal everything, okay? As long as you understand how the cycle works, that should be okay. Right, kalau tak ada apa-apa, kita tutup kita punya lecture lah dengan tasbih kafarah dengan surah wal asr. Okay, see you next week. Alright, bye bye.
Okay,
Assalamualaikum.
Malam. Thank you, Doktor.
Thank you, Dr.
Bye.
Thank you, Doctor.
Alright, bye bye.